Hey everyone, welcome to our Research America Alliance discussion this morning. I'm Ellie Dehoney. I'd like to thank you for joining on Zoom today and for your partnership in the Research America Alliance. If your organization is not a member, I hope you'll shoot me an email or Joel, whose email should be in the chat, or we're putting it in the chat now. Let's talk about membership and what it means. It would be terrific to have you join us. Speaking of terrific, I am so thrilled to introduce Dr. Jeffrey Cummings from the University of Nevada, Las Vegas. We've asked Jeff to discuss the science that has evolved significantly behind the new Alzheimer's drugs and how that evolution bears on, the pretty fraught debate, frankly, around these drugs and Medicare coverage. So as always, if you wouldn't mind typing your questions into the Q&A box or the chat, we'll definitely pose as many as we can during that portion of this discussion. And with that, let me turn over to you, Jeff, and thank you so much for being with us.
Thank you, Ellie. It's a great pleasure to be here. This is such an important moment in the history of Alzheimer's disease and in therapeutics for neurodegenerative disease in general. So it's really great to be able to talk about this. I'm a great fan of Research America. We advance our lives through research and understanding, and that's also why I've called my center the Center for Transformative Neuroscience, is because we believe that neuroscience transforms lives. And I think that's the philosophy that you share as well.
Beautifully.
I'll be talking about anti-amyloid monoclonal antibodies and how they have transformed care. Here are my disclosures. I consult for a lot of companies in an effort to find best possible treatments for Alzheimer's disease. I'll talk about the history of Alzheimer's and amyloid research. This has been a controversial area of Alzheimer's disease research. So I'll tell you a little bit about the controversy and also about our understanding of amyloid in Alzheimer's disease. I'll address the question, what are anti-amyloid monoclonal antibodies? How did we get to immunotherapy? It's a really interesting story, and it's about the march of research and the constant improvement in our understanding. And then I'll finish by looking ahead at what's next, because we're not at all at the end, we're at the beginning, and it's really exciting. So here is Alois Alzheimer. This is 1906. He described the brain of a youngish woman in her 50s who developed an unusual disease with dementia and psychosis, and he examined the brain. And he saw the neurofibrillary tangles, but he also saw something called miliary foci, and those are amyloid plaques. We didn't know it at the time. His boss named the disease in honor of him. How great is that? I wish somebody would do that for me. Then there was a long period of time in which there was no real progress, and this was regarded as an extremely rare disease. And then Sir Martin Roth discovered that really, if you look at the pathology of senile dementia, a common concept at the time, it was Alzheimer's disease. And it began to dawn on people that there wasn't something called senile dementia. There is Alzheimer's disease. These initial observations by Sir Martin were extremely important. Robert Katzman then in the US noted that Alzheimer's disease is common then in aging, and we're facing a tsunami of aging, and we're going to have a tsunami of Alzheimer's disease. And that was a 1976 observation and informed our thinking going ahead. Then George Glenner eventually just found that amyloid was the substance in the vessels and in the plaques. And so then we knew what the protein was, and that was a great step forward. And then there were a couple of steps to the amyloid hypothesis. People said, well, where does amyloid fit in the disease process? Originally, 1991, John Hardy and Alsop said, well, it's at the beginning and the rest of it follows. And then that was very much filled out by Hardy and Selkoe in 2002. So Dennis Selkoe and John Hardy are seen as the authors of the amyloid hypothesis. So here's what the amyloid hypothesis looks like then, that the first thing that happens is A beta amyloid protein accumulates in the brain. And that leads to the neurofibrillary tangles. And that leads to inflammation. And that leads to neuronal death. And the dementia is due to the neuronal death. So we think that amyloid is the provocateur in terms of starting the disease. And you see this illustrated a bit on the left, where amyloid is processed through enzymes. It then is generated, where it goes on to form plaques and to initiate tangles, to kill cells, and to create transmitter deficits. So why does amyloid occur in the brain? There is evidence now that we have clearance mechanisms because we all have amyloid, and those clearance mechanisms are compromised in some elderly. And when that happens, they eventually develop Alzheimer's disease. So we have some sense of why some people in the older population get Alzheimer's disease and some do not, and there are a variety of genetic influences on that, as well as aging influences. So from 2003 to 2016, there were many amyloid trials, and they failed 100% of the time. Now people say, well, why did you keep doing this? Why did you keep beating your head against the wall if 100% failed? And one perspective that's very important on this is that 100% of non-amyloid trials also failed. So it's not as if we were alone in the amyloid hypothesis-directed trials in terms of failures. There were also many failures in the non-A beta directed. All the trials failed between 2003 and 2016.
Jeff, can I ask a question about the trials themselves? I'm sorry to interrupt, but were all of the trials testing something that you thought could clear the amyloid plaque, or what was the nature of those trials?
They were all sorts of different approaches, Ellie, and there still are quite a few approaches in the pipeline. So some of them were directed at decreasing the production of amyloid. Some were aimed at decreasing the aggregation of amyloid into the plaque. And some were directed at plaque removal. And this was part of the learning. Research America is all about learning. So part of the learning was that we discovered that there were many species of amyloid.
Wow. And
you see that illustrated there on the left, that the first thing that is generated is a monomer, so a single molecule of amyloid. And then that aggregates into dimers and oligomers and eventually into protofibrils and fibrils. And then the fibril forms the plaque. Well, each of these is a potential target. And each of these would have a different different biology and a different pharmacology. And what we have gradually learned is that the closer you are to the monomer, the more likely the drug is to fail. And that the drugs that have succeeded are directed at the plaque and at the protofibril.
And the failure and success is not defined by clinical benefit. It's defined by what happens to the plaque at this stage, right? Is that right?
I would say
just at this.
We want both. We want the biology, but the biology makes sense only if there's benefit to patients. And I'm going to get to that in the monoclonals, because we do have both in the monoclonals. But the choice of your target determines the kind of trial you will do. And so that's why there were these different targets that evolved.
Makes sense.
So a very important piece of technology evolved that turned out to be very important. And that is the amyloid PET. And again, Research America is all about what's the advances in the research that has helped us to advance the science. Well, here's just a wonderful example. that amyloid, we knew amyloid was in the brain, but of course, we couldn't see it until the patient came to autopsy. And I always found that to be uncomfortably late in the doctor-patient relationship.
Yes.
So is there a way to see amyloid in the living person? And sure enough, there is. And this was first published in 2005 with Pittsburgh compound B. And then that evolved to compounds that are much easier to use and to generate. And that was the first approval then was in 2012 by the FDA. So there are 3 approved types of amyloid imaging. And then they all show the plaque. And what I'm showing you on the left there is 3 vertically, you're seeing at the top an image of that it's normal, in the middle an image that has a beginning signal, and in the bottom a full-blown, that red signal is the amyloid signal. And you see then in the corresponding histological sections, no plaque in the upper panel, a lot of plaque, but an intermediate level in the middle panel, and a huge number of miliary foci, as Alzheimer called them, in the lower panel. And this was the way that we knew that amyloid PET was showing us amyloid in the brain. The FDA said, okay, that's what your claim is. You have to scan patients that then die, and you show that scan predicted either negative or positive evidence of amyloid in the brain. And so patients were scanned at very near the time of death, and to show exactly what I'm showing you in the left set of panels. That technology is at the basis of much of what we're seeing now.
It's amazing.
Now, what about immunotherapy? I mean, nobody had thought about immunotherapy before 1999 and Dale Shank's seminal observation. We all thought about small molecules, or the field thought about small molecules. You know, how do you give a pill? How do you interrupt this process? And nobody thought, well, could you use a vaccine? I mean, vaccinate against a neurodegenerative disease? How
Right.
an idea is that? Well, that's the kind of wild idea that takes root. So Dale, who was a personal friend, and I'm sorry to say succumbed to pancreatic cancer after this discovery, you see that he immunized with a vaccine, and the vaccine was aimed at amyloid. And you see in the upper 2 panels an unvaccinated mouse, and all that red material is amyloid. And you see in the lower panel a vaccinated mouse. And you see that the amyloid has been removed. And this was a revolutionary observation. I mean, just way out of the box, way out of the hemisphere of our thinking. And so that was the beginning of immunotherapy then. And Dale suggested at the end of this, maybe this is a new way to approach Alzheimer's disease therapy. Notice the way that it takes until 2021. So it takes 22 years to get from this observation to the first approved therapy by the FDA aimed at amyloid. So what was the story then about the emergence of amyloid? Well, there was a human vaccine trial. These mice were vaccinated, so let's do a human vaccine trial. Well, that was called AN 1792, and 6% of patients developed an encephalitis. So not only did the vaccine stimulate removal of the amyloid, but it also stimulated an adverse reaction, and 6% is way too much, way too much. It is, by the way, why phase 1 and early stage patients are heroes, because we don't know the side effects of these drugs when we start working with them. That's the nature of clinical trials. You have to recognize that. So then people thought, well, maybe if we can't give a vaccine, maybe we can raise the antibodies externally in cells and then infuse them. And that's monoclonal antibody therapy. So that's what is passive. And the first drug was bapineuzumab. It was published in 2014. I have the references there below. And it failed. And then other antibodies also failed to show a drug placebo difference, but none were directed at amyloid plaques or the protofibrillar amyloid species. So none had the target that we were evolving towards in terms of our understanding of the biology. Also, these studies suggested that the dose of the monoclonal antibody that we were using in these early trials was way too low, 5 times too low. So the doses that we're using now are 5 times what they were for bapineuzumab. And it's highly likely, actually, looking back at bapineuzumab, that if we'd used the right dose, we would have discovered this in 2014.
Wow.
That's part of the evolving process, right? You don't
Right.
know the dose when you start. This was, and I'm showing you there on the right, by the way, the encephalitis that was seen by AN 1792. You see that white signal on the MRI. So it was a serious event, and that's why the trial was stopped. So
Jeff, there was one point that was made, I'm so sorry, in the last slide.
Sure.
It said one of the observations was dose too low. The other observation was that plaque removal is important.
Yes, we began to suspect that from the bapineuzumab studies, that we were going to have to address the higher level species because they were the more toxic. And the drugs that were failing were directed down at the single molecule of amyloid level. So our knowledge of the toxicity of amyloid was growing. And that led to this turning point in 2016, which was the PRIME study. This was aducanumab. It was plaque focused. So this was a drug focused on the plaque. The dose range was explored, including to a high dose. In phase 1, the high dose produced marked amyloid reduction. First time we have seen that. And the clinical measures, although they were not uniform in a small trial, supported that there was a clinical benefit as well, that there was slowing of the disease. So and let me show you those scans on the right there. The 2 upper scans, that red signal is the amyloid. And you see baseline and 12 months, the upper two scans are on placebo. There's no change in them. The lower 2 scans, you see a red signal at baseline, and then no detectable red signal after 12 months of exposure. Undetectable. We've never seen a change like that, from detectable, robustly detectable, to undetectable in terms of the proteinopathy that is in the brain. And that's what PRIME showed us. So that led to phase 3 trials. And unfortunately, there was a misinterpretation that these trials were futile and they were terminated. But as additional data came in, it was clear that one of the studies had been positive and one was negative. And even in the negative study, in the people who got the highest dose for the expected period of time, it was positive. So actually both trials were positive for the expected dose, for the expected exposure. And the FDA by then was also beginning to see information that the rest of us didn't have from other monoclonal studies. And so they provided accelerated approval for aducanumab.
And when you're saying, I'm sorry, positive versus negative, do you mean positive for plaque removal or negative for plaque removal or for clinical benefit? You said in the phase 1 trial, the clinical measures were not, it was too small to, you had variation in the clinical measures. Is that plaque removal or is that actual clinical benefit?
So the plaque removal was marked in both studies.
Okay.
See that we're really doing that. So, but you could see the clinical slowing in the Emerge study, and you could not see the clinical slowing in the Engage study.
I see.
Unless you looked at the group that got a high dose for 14 months. And then you could see the slowing in Engage also.
I see, but that was a small
That was
That was a
the
small group.
but
Okay.
it was enough, along with other data that the FDA had to provide accelerated approval. And this
I
was very
see.
controversial.
Okay.
It's the greatest fracture in the Alzheimer community that has
And it
occurred.
still exists, right, Jeff? There are those that still are conflicted.
So there are, although with lecanemab now, and I'm going to show you that in a minute, there's far less controversy because the outcomes are absolutely clear now.
Oh, terrific.
There's an ongoing argument about, are they enough? And that's an important argument as well. There's debate, I would say. But there's now no controversy that these drugs produce slowing. And that was, I just want to call attention to this. I want to take you back to those histological sections I was showing you with the negative and positive scans, and then show you a pre-treatment scan and a post-treatment scan for lecanemab, and look at what you are removing. You can see that you're changing that bottom histological segment to what looks like the top histological segment with no detectable amyloid.
Wow.
It's really, to me, it's just a breathtaking observation.
Yeah, absolutely.
It's amazing because we have had no disease-modifying therapies for Alzheimer's disease. This is a disease-modifying therapy, no question about it. And we have very few disease-modifying therapies for any neurodegenerative disease.
Right.
ALS being the exception. There are 3 drugs approved for ALS, but that's the only other neurodegenerative disease. And think how common Parkinson's disease is. And then there are many rare neurodegenerative disease. None of them have disease-modifying therapies, but now we have it for Alzheimer's disease. So this is a change in the brain that's occurring. So now we have 2 studies, and this brings us now up to date, Ellie, that we have the lecanemab phase 3, marked plaque lowering to below detectable levels, positive on the primary clinical outcome, which was a CDR, sum of boxes, the clinical dementia rating, sum of boxes, positive on all secondary outcomes of cognition and function, and positive on exploratory outcomes like quality of life. So this led to accelerated approval based on the phase 2 data, and standard approval is now being considered by the FDA. The advisory committee is this week.
Wow.
No, is it this week? Yeah, this week or next week. And we expect the news on July 6th. So we're within weeks of getting standard approval for lecanemab. The FDA came out yesterday with its analysis, and its analysis looks positive for a standard approval. We'll see. It hasn't occurred yet. The donanemab phase 2 study has been published, marked amyloid lowering. We see that now we can do this. Positive on the primary clinical outcome, which is what matters. It was not positive on the secondaries, nor was it powered to be positive on the secondaries.
And when you say powered, does that mean
you have to have a certain number of patients in the study?
I see.
In order to see a drug placebo difference. And so you determine the number in a phase 2 study, you determine that number based on your primary outcome.
I see.
Therefore, you're not likely to have enough patients to determine less sensitive instruments.
I see.
And that's exactly what occurred. But they have already had the press release on their phase 3 trial, and there all the primary and all the secondaries are positive. But we haven't seen the publication yet. So we're still waiting for full peer review on that. But what we can see is that both of these drugs produce marked plaque lowering, and both of them have clinical slowing. Now, nobody gets better. This is very important for patients to understand, for doctors to understand. Nobody gets better. We are slowing the progression. We're slowing it by about 30%. So people they have an additional year or 2 in the mild phase of the illness before they progress into severe dementia. That's very important from my point of view. but it is a matter of controversy as to whether or not that's enough. So that's where we are in terms of these particular drugs. Now, there's an important side effect of monoclonals, and I should say that monoclonals are delivered intravenously, so this is not convenient. They're delivered intravenously. With lecanemab, the infusion is every other week, and with donanemab, it is once a month. And a few patients get something called ARIA, amyloid-related imaging abnormalities. It's thought to result because we remove amyloid not only from the plaques, that's our intent, but there's a lot of amyloid in the blood vessel. And when you remove that, when you put the monoclonal into the blood by the infusion, it pulls the amyloid out of the blood vessel in addition to out of the plaque. And that causes the vessel to leak. And so you get blood or fluid in the brain. That happens in 15 to 30% of patients. Most of them are asymptomatic. 3 to 5% have symptoms. So most people have it, and who have it do not know they have it. But it's important for doctors to know, because when that happens, we stop therapy unless it's very mild. And then every month we get an MRI scan, and then we go ahead to give the, sorry, my dogs are barking in the background.
No, no worries.
We interrupt therapy until we see that the ARIA has gone away, and then we recommence therapy. Rarely this can be fatal. That's why we have to monitor it so that we can stop. There's been about 3 cases of lecanemab where there were fatalities. 80 to 90% recover and can be continued on therapy. So it must be monitored with periodic MRI. And in the series of MRIs below, I'm showing you on the far left, a normal scan. Person comes in, gets his first dose, develops ARIA pretty much early on, within the first few months. That's that white signal at the back. It gets worse, then it begins to recover, as you see. And by the end, he's actually back to normal, back to where he started on the MoCA score, which is one way of scoring cognition. So you can see that this recovery is complete, but you also see that's a pretty nasty looking lesion on the MRI scan. And that's why we stop therapy, so that we make sure that we don't exacerbate that more. So these are complicated therapies. It's not surprising. We're thinking much more like cancer now.
I was going to ask you about that. Is there a way, in relation to other infused drugs for rare diseases, for cancer, are side effects like this, potential side effects like this, and kind of this intensity level, common, not common? I mean, is this a kind of a black box warning type of a problem, or, you know, can you put it in context for us at all?
Yeah. Well, I think we've come to understand that cancer drugs do have a lot of toxicities, actually much more symptomatic toxicity than the monoclonals do, because you see only 3 to 5% of patients actually have symptoms with this ARIA, and there are very few other symptoms that occur with monoclonals, but they're serious. And that's why it's so important for us to monitor it, because they can be serious. So it's hard to sort of compare it exactly with the cancer therapies.
Yeah.
Certainly much more like cancer therapies than the pills we have had so far that have been so benign, but also are not disease modifying. So this is the opportunity to interrupt the disease, but it also has complications that must be attended to.
Excellent. Thank you.
So this is an unprecedented therapy, and therefore it makes unprecedented demands on healthcare systems. And that's the discussion we're having now. So first of all, only early Alzheimer patients were included in the trials. So we think only early patients should get these drugs, because that's the only group of patients that we know have efficacy and where we understand the safety. So they have to be recognized. Right now, we know most patients aren't recognized by their doctors. They're excused as normal aging. So we have to increase the vigilance of doctors and families, for that matter, for the importance of early symptoms. It then has to be confirmed by either lumbar puncture or an amyloid PET, though I'm going to tell you in a moment about blood tests that look so good that I think we'll have them soon.
Wow.
Patients must then have an MRI prior to starting therapy to make sure that there's not a lot of blood vessel disease in the brain that's obvious at the beginning. Treatments are given intravenously, as I mentioned, once a month or twice a month. MRI must be obtained periodically during that initial year. All of those ARIA, not all, the huge majority of ARIA, 90% plus, occur in the first year of therapy. That's when the blood vessel is kind of adjusting to this new state that they're in. Then the blood vessels recover. ARIA can occur, and reimbursement by CMS is uncertain. It seems increasingly likely that they are going to reimburse these. We'll know soon, but there's likely also to be some sort of framework around that reimbursement that we'll have to understand much better. So where are we now? Anti-amyloid therapies slow the progression of AD. Hallelujah.
Yeah, yes.
This is a really a great breakthrough. For me, this is the most important thing, that we show that we can slow the disease. Now we can do a lot of other treatment, right? This is what Research America is about. Let's take the next step and the next step and the next step.
Right, right.
And we can build on this observation. So these agents are disease modifying. That's the breakthrough. They've been approved by the FDA by accelerated approval, and we think standard approval is coming probably within a month. They require infusion MRI monitoring, and they have side effects we have to be aware of, and reimbursement is currently uncertain. We're trying to understand that. What's coming next? I think subcutaneous administration of monoclonals is highly likely. And that will make it so we do not have to have infusions in infusion centers and all of that. I think diagnosis by a blood test is likely within 2 years, maybe sooner, because we already have blood tests that are performing very well. We just have to show that in a whole variety of circumstances, they continue to perform very well. Right now, we have the blood test on very well characterized patients, but we need to understand it in the general population where they will be used.
Hey,
but
Jeff.
Quite close.
Forgive my ignorance. Is subcutaneous like a patch? Or?
No, it's more like getting a TB test, where you get a little, they take a needle, put a subcutaneous injection, it raises a little welt, and in that would be all the antibodies.
Okay, perfect.
So it would be subcutaneous and not cutaneous, like patches.
Perfect.
So we're likely to be able to do without PET very soon. Other types of treatment are also going to come along. That's the great power of an advance in disease modification. And we already see that something called ASOs that interferes with the gene translation in Alzheimer's disease looks so positive. Just really exciting. Now that drug's also complicated, has to be given into the spinal fluid. So that's another complication, but you'd only need it every 3 to 6 months. Anti-inflammatory agents, there's a whole bunch of these in the pipeline, about 20. And we know that there's a lot of inflammation in the brain. So they might be helpful. They also might be helpful in decreasing ARIA, because ARIA has a lot to do with amyloid. There's a bunch of synaptic agents that might have the benefit of improving cognition as well as slowing the loss. That would be terrific. Patients want to get better. I want them to get better. And we would then use a drug that slows cognition and a drug that improves cognition. That'd be a great combination. And we're going to have combination therapy, as we do in AIDS and cancer and tuberculosis.
Yeah.
And metabolic agents derived from diabetes are also making a lot of progress at this point. So this has been a great demonstration of research discovery leading to new therapies. It's an illustration of the importance of breakthrough technology, amyloid PET. It reveals a need for a healthcare system that is planning to incorporate unprecedented therapies. This is not the last unprecedented therapy. We're going to have lots of breakthroughs. We're starting to understand the brain in a unique way. But we have to be able to take advantage of the advances by a healthcare system that has a plan for incorporating new therapies. We have to do that. And I say science forward. Thank you.
What an absolutely terrific presentation, Jeff. I learned so much. I think we all did. I do think, I think you hit on kind of the fundamental tension here. And that is that that medical progress is taking us to a place that requires that affects the health system in important ways, and we've got to reconcile the two in a way that doesn't slow progress. But we've got to recognize that these changes are difficult, and you know, and that our federal regulators and HHS are struggling with some very hard questions. But it bears on progress across all diseases. There are a lot of great questions I see here in the chat and in the Q&A. I don't even know where to start. So Jacqueline or Sam, if one of you would join us and start at the you know, people who have been waiting the longest, that would be great. I think Nancy's questions were answered. Cliff, I think your questions were answered. But anyway, if you could keep on going down the list, Jay, that'd be great.
Sure. Thank you, Dr. Cummings, for this fantastic presentation. First question from the audience. Can you comment on the general concept that Alzheimer's disease and other neurodegenerative diseases likely begin years or even decades before symptoms begin, and how that influences treatment effect? In other words, how likely are even effective treatments to work when started years after amyloid begins to deposit? Should we begin treating much earlier based on imaging findings or family history, etc.?
But a great question and a very informed one, so I can tell whoever answered that knows this Alzheimer literature. Yeah, this was another observation that was derived from the advanced technology. When we had the ability to image amyloid in the brain, we could certainly see that it was present for 15 years before symptoms begin, 15 years, and about 20 years before dementia begins. So this is resident in the brain a long time, and it's surprising that removing it after 15 years still has a measurable clinical benefit. But the great thing is, since we can see the amyloid in the brain, we can also do prevention trials, right, before people get symptoms. Wouldn't it be great if we didn't have Alzheimer's disease?
Right.
So now we can see who has it in the brain. We might even be able to see who's at risk for getting it in the brain. So there is a huge amount of attention right now to prevention studies. Lecanemab and donanemab are both in prevention studies already. So that's going to happen. We're also moving towards vaccine therapy, back towards Dale Shank's original observation that we can probably safely do vaccines. And it would be really much better if people just got a vaccine, even if they needed a booster twice a year, because the older the immune system is, the more difficult it is to get the response you need. But wouldn't it be great if people just got a vaccine twice a year and didn't get Alzheimer's disease? That's where we want to be. I think we're coming closer and closer. So I completely agree with this question. We can detect it early, it's in the brain early, and we want to move early. But I also feel very strongly that we have 6.5 million people in the United States with Alzheimer's dementia. We can't forget them. No. We need therapies for patients who have symptoms, in addition to those who are at very high risk for getting symptoms.
And Jeff, I want to just clarify that when you say prevention right now, it still would be removal of plaque, not prevention of plaque, right?
So for the current trials, that is true, but we're already conceptualizing primary prevention. which is identifying people who are at a high risk and beginning them where the outcome would be the delay to plaque occurrence.
Really? The same drugs could have that effect?
Yeah, same drug could have that effect.
Okay.
That's where a vaccine would make a lot more sense, right?
Yes.
Rather than an infusion when somebody who's many years before the onset of the disease. But that's where the vaccine would really make a lot of sense. And that's why we think that the vaccine strategies are also very important in our field.
Excellent.
Great, thank you. Next question. What is your opinion about the advantages, if any, for targeting hope I pronounce this right, oligomers over monomers?
Ooh.
Yeah. So that's also a very sophisticated question. I love your audience, everything we're talking about. So we have discovered that I've mentioned the different species of amyloid, and it starts with one molecule of amyloid being produced, and then they begin to stick together, and then they form the plaque. Well, that sticking together is the oligomer, and at least in the laboratory, the oligomer is the most toxic species, more so than the plaque. So a drug like lecanemab, and actually also aducanumab, target the oligomer as well as the plaque. And then donanemab is focused on a species of amyloid that is only in the plaque. So we're in a very good position to understand, does targeting the oligomer add something to removing the plaque? So we're going to just learn a ton about this. Again, we're at the first step of this. We're babies in terms of trying to understand this. So the oligomers look like they're important, and we're positioned to understand whether they are important.
Great, thank you. Next, do we know how expensive these blood tests would be? In other words, would it eventually become part of an annual physical for older adults?
Absolutely. I think just like men get a PSA when they go in now, I think we'll want to do this with all people beginning at the age of 50, say. They'll be very financially reasonable. We already have competitive blood tests in the market. They're not approved for diagnosis yet, but they're already there in the market. So I think that will drive the price down. Right now, it's about $3,500 to get a PET scan, and about $1,500 to do a lumbar puncture, maybe $1,000 sometimes. We should be able to do, for a few hundred, we should be able to do the blood test, maybe even less. So this is going to be a dramatic change, both in terms of our ability to detect persons when they can benefit. That's the important thing. And starting therapy. And it looks like they're going to be able to monitor therapy as well, because these blood tests change as you remove the amyloid from the brain. So we should be able to detect and monitor with the same blood test.
Jay, let me interrupt. I'm going to go to Matt's question, but before that, really quick, I think one question was from Mitchell, and he said, do you treat ARIA with steroids, or does it dissipate on its own?
Yes. So it depends on the severity. It's a really good question, again, from a clinician, I can tell. So if the ARIA occurs and they have no symptoms, we would not use steroids. But if it has symptoms, particularly if the symptoms, if they had a seizure, something like that, we would definitely use steroids. And I work with a group that's produced something called appropriate use recommendations. And in the lecanemab appropriate use recommendations, we describe exactly how to give steroids, what doses. It's a hands-on, step-by-step. Here's how you do the MRI. Here's when you do the MRI. If you see something, here's what you do. The appropriate use recommendations are a sort of guideline. That's too strong a word. They're recommendations for how you handle these drugs.
Great. Matt is getting into the coverage controversy in an excellent question, but he is saying that it's his understanding, I believe, Matt, if I'm catching this right, that as it stands, CMS has said they will cover the drugs, but under pretty constrained conditions in which patients have to be part of a registry. I'm not sure what the other conditions are. So, Jeff, I wanted to just make sure that you're comfortable with what the actual state of play is in this coverage debate. The
Yes.
distinction between just an outright, listen, this is reasonable and necessary. We are going to, for certain population early on, early in their phases, versus what CMS has said they are willing to do for drugs in this class.
Yes, so I'll say what I know, and if Sue wants to expand on this, she's in the audience.
That's true. We have Sue Passion in the audience, so we have a resident expert. That's awesome. And Ian and others.
So let me go back a moment to accelerated approval of aducanumab. That was on the basis of plaque removal, and there was controversy, as I outlined, about whether there was clinical benefit, with one negative and one positive study. So in that circumstance, CMS said, yes, we'll cover it, but only if the patient is in a clinical trial, because we need more data. Well, that's severely limiting. And essentially stopped the further development of the drug. But with standard approval, which we think will occur very soon with lecanemab and is likely to be followed by donanemab, the CMS has so far said they're going to use a registry, and they've used words like a light touch registry. That's what the CMS administrator said in front of Congress, light touch. Well, that's masterfully vague. It sounds good, but we don't really know what it means yet in terms of what kind of demand would it place on physicians to register a patient, and what kind of demand would it place on the patients? Would there be extra requirements? So we're trying to understand that, and there are a lot of ongoing discussions, and there are probably, I'm sure there are people who are more up to date on what that might look like. But we do believe that it'll be far less restrictive than being in a clinical trial. How much less restrictive, we don't know.
I think the tricky thing here is, and I know that CMS is working in good faith, but the tricky thing is the why. What problem is the registry going to solve? And my understanding is that this coverage with evidence development model, CMS does not acknowledge that these medicines are reasonable and necessary, and it's kind of getting at the thing. What the registries would be great for every single drug that is approved, obviously, but I think that's what's so difficult from a patient perspective is, why are these special requirements being placed on drugs that are showing clinical benefit? Is it because it's so tricky to understand the significance of the clinical benefit? Or, I don't know, Jeff, if you have any insights into this, but I guess that's my question about it.
So I mentioned that these are unprecedented agents, so they pose new questions for us, and CMS is aware that by the time you pay for the drug, and right now a PET scan or a lumbar puncture and the MRIs, you have a substantial cost, and there are theoretically a lot of people who would be eligible, although the analyses show that if you match the trial population, there actually aren't so many. So the benefit, the financial impact may be less than many people declare. So that's something we'll come to understand. The larger discussion is, what is the value of cognition in older adults?
Right.
How much are we willing to pay as a society for people to spend a year or two longer with their families with reasonable cognition? And that's a hard question. I think my leaning is, since the Enlightenment, we have wanted progress that would improve human life, and I would include human cognition as the basis for that. but there's a financial argument to it, and some people value that more, and we do have to solve the finances. That's why the progress towards cheaper treatments that are effective is so important, right? This subcutaneous administration with a blood test for detection,
Absolutely.
a whole different package, right?
I also think from CMS perspective, I can understand that this is a new thing to think about in terms of reasonable and necessary for them. I mean, I really understand they're afraid of false hope being created, that what does 30% feel like? What does it look like for different patients? But I think that that's almost a question that, for one thing, patients really need to be involved in that answer. And it is a system-wide question, but we can't ignore. It's really important to separate progress from the questions that arise from it, as I've said. So I'm kind of with you in that we need for this to keep happening. We can't stop it because there are hard questions around it in the health system.
That's exactly right. Another part of the formula, Ellie, might be that 30% slowing is the mean across the whole population of 1,000 people, or 500 on therapy. Well, there were responders and non-responders in that. And if we could identify the responders, we could treat fewer people, because there's no reason to treat a non-responder.
That's interesting. I didn't realize that.
So another part of this is, again, more research, is to understand what determines whether someone will respond to therapy or not. We know almost everybody gets plaque removal. Some of them then get the slowing, some very substantial slowing. 50% didn't change at all in a year.
Right.
Amazing. But who were those 50%? You know, right. What were their features? What were their characteristics? Can we identify them ahead of time? Those are the kind of questions we now need to probe.
And I think, though, in the broader argument, that the fact is that if this becomes a prophylactic, if we were able to prevent, you know, I remember when a doctor said, you know, we should just put statins in the water, right? You know, so many people need statins, but the fact is that they're pretty affordable, right? And so this is a very big system disruption potentially, as if it builds out, if the science builds out and builds forward to folks that have more dementia, but maybe we can help them. It becomes a big cost and systems change question, but we just have to confront that. We can't hide from it by slowing the progress.
Right. That's why the dialogue and all the stakeholders have to be in the dialogue.
Yes, exactly right. Exactly right. Oh, we have very little time. I've been blabbing. Other questions in the chat. Jay, have you seen?
Let's see.
I'm not sure, Mitchell, if we can go to putative vascular contributions, only because of us non-docs in the audience, but
I'll just comment. We know very little about the vascular contributions because patients with substantial vascular disease are excluded. This is another problem we have to solve. We're excluding too many people. And when you exclude vascular disease, by the way, you disproportionately exclude Black and Hispanic individuals.
Oh dear, from the trials.
Because there's more hypertension, there's more diabetes, and therefore there's more vascular disease in minority populations. So our exclusion criteria disproportionately penalize our minorities, and we just can't have that. We have to figure out a way to manage that.
What's the path that's being taken forward to address that?
Massive attention to why we're doing so badly in terms of incorporating more minority individuals, because of course there are plenty of healthy minority individuals too. Why can't we find them? You know why?
Yes.
You know why is it, why are we struggling with the seclusion problem? So we just have to do a much better job. We're really trying to figure that out. There's a lot of new grants that the government has provided in order to try to understand minority recruitment. It's a major commitment. We have to have equity. We can't have therapies just for white people.
No. And
So this is something that we have to confront. It's another part of the dialogue.
And are folks trying to figure out the vascular effects so that maybe they?
Yes, yes, we would. There are many avenues to that, but yes, there's a lot of attention to that, and some other exciting science that I didn't mention about how to get antibodies into the brain better, and then maybe we would have to be less exclusionary in terms of who can get them, and so there are other avenues that are being investigated.
That's just terrific. And I have to apologize to Mitchell. I'm so glad, Jeff, that you jumped in. I got so scared of terms I didn't know, and that is not fair. Just because I didn't go to med school does not mean great answers to great questions can happen on this webinar. I think we are basically at time, unfortunately, and the participation on this webinar has just been fabulous, and I think it speaks to our wonderful speaker. Jeff, we could do this for 3 hours, and I think it would be, no one would be bored. It would be, we learned so much in such a short span of time. Folks have asked if they could get a copy of your slides. Is that good by you?
Yeah, absolutely. We just think about whether there's anything there. No, I don't think there's anything there that, that can't be shared. So I'm happy.
Okay, and we can, we'll do a little space of time in case there's anything needs adjustment. And also the recording is going to be available. Great. And Jacqueline, could you quickly talk about, I know we have an Alliance member only meeting coming up, and.
Yes, so if your organization is a Research America member, join us next Monday at, no, I don't have the time. It is at 12. So we have James Lucier of Capital Alpha Partners talking about the implications of the debt deal for research, public health, and public-private R&D partnerships. He'll be answering what is the outlook for funding for NIH and other research and public health agencies. What does the deal mean for pandemic preparedness? What can advocates do to help shape the answers to those questions? And does the deal affect other legislative efforts like reauthorization of PAVA? Please bring your questions and insights, and I'll drop my email in the chat. If you are a member and haven't received the invites, you can shoot me an email.
And if you're a non-member and want to join us, give me a call. That's not how you do give me a call, but you know what I mean. Get in touch. Yeah.
Thanks everyone for joining today, and we look forward to seeing you again soon.
And thank you, Jeff. You were just wonderful.
My pleasure. My pleasure. Thanks for including me. Bye.
Take care, everyone.